All right, so I'm going to talk about data visualization principles. I'm not going to teach you how to make the plots, but this is a Quarto presentation, and all the code is here. I'm not showing it, but it's in the file. I'm going to share it at some point. I don't know if we had it already, but if you have the presentation, the code is all there that makes this presentation. So you can see the code that I use to make the plots, the visualizations that I think are appropriate.
I just shared the link in the chat for anyone who's on Zoom.
And this one is not. This one's not completely reproducible because it loads a data set that we haven't shared yet. We could this one data set at the end of the talk, and I think there's like 3 images I need permission to share before it's public, so they're on my computer, not, and you won't, when you try to run it, you obviously won't get them. So the talk has a lot of images that are pre-made or just screenshots. Then there's a lot of plots that are made within the presentation. Okay, so I'm gonna so the approach that I'm going to use to teach you the visualization principle that I like is to mostly I'm going to show you bad plots and then how to improve them. So that's usually how it's going to go. So I'll show you not just bad, but a bad plot that's made very often, and then show you what we think is a better way. And then just to say that a lot of these come from this a lot of the examples come from this blog post, and also from Carl Broman. I thought I had that here. No, Carl Broman has a chapter on this too that I borrowed. Okay, so I'm gonna start just with some motivation of really bad infographics. It's mainly for fun, but a little bit for motivation to see how deceiving plots can be if made in a way that's made to be deceiving. I have some really ridiculous one like this one. Make it look, just take a little bit to notice the issue with it. Here's another one. This is from the US news place. The y-axis is on going up. Yeah. This is another great one. Here's another one that's doesn't add up to 100. This one is interesting. They're showing the increase as the y-axis, not the actual value. And this is very common. I'm going to be showing this more later. This is a very common error that even in science journals you see them very often. The y-axis is not starting at 0. And then this is another example of this is my personal favorite. It shows you the difference in size of height and women in Latvia versus India. You see that the little there is the heights of the heads is correct, and that's the correct average height, but it's not starting at 0. And also this one has 2 problems that they also use area instead of a linear area, so it's squared. It's the height squared, and it's very deceiving. All right, so now with those ridiculous plots out of the way, we'll get to some more serious ones. So here's I was mentioning that a lot of this comes from Carl Broman. Sorry, at the beginning when I said a lot of that blog post was about the ridiculous plots, these 2 links take you to the material where I borrow a lot of the ideas of visualization principles, so more serious stuff. And if you download download this as an HTML, you can click on it and go to these links. If you have the code, it'll be right there for you to copy and paste. Yeah, so just I don't know if everybody online, there's a lot of more people today than usual, so I don't know if you know what I mean by Quarto, but it's code. So this is the presentation in Quarto code. So it makes the presentation and it includes code. So you can see an example there that makes the plots that you see in the presentation. So you can see what a plot you like, say, how do I make that? Go back to the code and grab the code from there. Okay. How do I get back? Yeah. Okay. All right, so let's get started. So again, I'm going to start by the whole presentation, which is going to be about 2 hours. We're going to go through examples to show different principles. We'll start with a bad one just to motivate the correct visualization approach based on actual principles. So I'll describe the principle, show you a bad example, and then show you how I would fix it. Okay, so here is so when why are we making plots? So this is one of the first principles. Is like you want to know why you're doing it. Why are you making a plot? We want to show numbers is a main answer to that, right? When you're showing data, a picture is definitely worth 1,000 tables and data points often. So it conveys the information you want to convey visually. It's faster. People get the idea quick. That's why we make plots. Today, I'm seeing in a lot of journals, especially in biology journals, some medical journals are also falling into this. A lot of the plots seem to just be art. They're just showing pretty things. And when you try to figure out what they're telling you, it's impossible. I'll focus on one of those. It's at the end, which are the UMAP plots. But there's others. There's other plots that are used very commonly just to impress with the visual effects, but they don't actually convey information. So that's the first thing to keep in mind. You are trying to show numbers through plots. That's at the end of the day what you're trying to do. And these are some of the tasks we try to do. We can describe distributions. We can compare data from two groups. We can compare relationships between two variables. These are all kinds of things that we try to show with plots. So to do this, we have available to us to show numbers as something you look at. There's 6 approaches you can use. And this is ordered from best to worst in how well humans, and this comes from psychology studies, how well humans can translate the visual cue to a number. Right? That's key. You want to be able to see something and if the number gets, you see the number in your head when you see the picture. So the best one is a line length. So if you see a length on a picture, you know how long it is. And if you see 2 of them, you know this one's longer than this one, right? That's very, very easy to see. The second best is position. That's points where you can see on the y-axis how big it is. So you have to do a little bit more work, but it's very clear what it is. Third is angles. I'll show you some examples of that. The pie chart is an example that uses angles. Area, fourth. Big an area is, we're not that great at seeing it, but you can distinguish something with big area versus small, but it's hard to exactly see what number to put with it. Brightness is next to last, dark versus light, and then finally the color. So I'm not saying color's bad. I'm just showing you the order in which us humans can take visual cues and convert them into numbers. So using this, we'll go through plots that for example, go straight to angles and don't consider lengths for the plot. And that's the first example is pie charts. This is all over the place because I think it's because Excel made it really easy to make. Must have been like 30, 40 years ago when pie charts started showing up everywhere. But this is the example. This is a real example. Or is it? I think it is. It's percent of users of browsers 2000 and 2015. So what are we trying to convey when we make a plot? First, we want to know what percent, which was the most popular browser, 2015. Another thing we might want to see is which one grew more than the other. But that's very simple. So how do we show that? So there's 5 browsers. Want to know how much each one is being used, and unfortunately, the default it seems in science when you have things that add up to 100 is to make a pie chart. It's not good. So if you look at, let me just show you. So in this plot, which is the browser that's most used? Which is the main thing you want to know? Can you tell? Which is the biggest angle? So here we're using angle and area to give a number. Right? So which is the bigger angle? Purple or blue?
I think it's the big one.
You think, right?
It's hard.
I will see soon. You know how we're gonna see it? By actually looking at the number. Which would fit in this space. And then which one grew more? You can't see it. It's not a good plot for showing numbers. It's very rare that a pie chart is ever the thing to show. Very, very rare. Except that, you know, the percent of sun on hitting the pyramid. Have you guys seen that one? Now I have to show it. This is a good use of a pie chart. This one.
Oh, yeah.
Yeah.
Okay. So there's another one that's like how much pie I ate, which is left. So
The one I saw.
oh, no, where did it go? Now I can't find it. Oh, I have to go back. There it is. Okay. Okay, so that's we're gonna see so which one did I say was the best way? Linear, right? So we're gonna make a linear plot. It's a bar plot. That's all you may need. In the case of 5 numbers, you can just show the numbers. But if you want a visual version, you use length. So this is worse than a pie chart. I think these are kind of going away. For a while, they were all over. All of a sudden, pie charts were out, and these donut plots were in. So what you do with donut plots is you take the best thing about the pie chart, which is that it shows you the angle, and you get rid of it. And now you're just stuck with area for figuring out which is the bigger one. So you went from having some information to very little. So here's how I would make it. This. There you go. And now we can answer the questions I asked. It was Chrome. I was Internet Explorer, but in 2015, Chrome took over. Okay. That's it. Now, if you want to show up a plot for this, which it could be better to show this. And this is length. And yeah, right away, IE is the highest. But then in 2015, it's Chrome. No, just look at this for a little bit and see how quickly you see it here and how hard it is to see here. All right, so that's my first example. All right, any questions, comments? This will also be easier on your printer. That one. All right, so now if your PI for biologists on Zoom, if your PI just insists, your boomer PI insists on a bar plot, then for the rest of us, so your readers can actually see what the hell you're showing, put the numbers there so we can actually know what's going on. We can ignore the pie chart and look at the number. Right, so that's a middle ground for those that need to be putting a pie chart, because it is like very strong. The momentum is strong with pie chart. All right. So when to include 0? Next topic. We saw an example of this in the ridiculous plots about a politician that wasn't including 0, so he looked like he was ahead by more. It's very common. You see it in science papers. You see it a lot in the news and in political ads. So if we're using, here's what it comes down to. If you're using length to show a number, if your visual cue is length, right? So bar plots are length, then you have to show the length. You can't cut it off. So that means you have to start at 0. Okay, so here's my first example. That's bad. Very common in the news. So you're seeing, it looks like in 2013, border apprehensions went up quite a lot, right? You look at because they're implying by using a bar plot, they're implying that length is the visual cue. So I look at this length, and that's about 2.5, 2.5 more than this one, right? But it isn't with the numbers. If I start at 0, it looks like this. That's the plot they should have shown. They shouldn't use yellow though. All right, so there's so many of these. I'm going to just show you a few. Here's another one. This is, again, 39 versus 35. They don't start at 0. Here it is starting at 0. This is my favorite because they actually made it into some kind of cylinder. Not just a bar plot. That one's fantastic. Like this looks like it's like what, like 20 times bigger.
It's such a loser.
Also the precision. So then that's what that one looks like. And here's a recent one. You just look, production increases. Look at that, almost doubled in 2025. No, it did not double, and this time Twitter or whatever it's X is telling us. Use X. They sometimes have what is it called? Community notes are really good, actually. I find them to be very good. Look at this. Looks like I wrote it. I didn't. That's exactly right.
Was that the White House?
Yeah.
Okay.
That's an actual White House tweet.
Yeah.
Cool. Oh, but there's not just a Republican. I want to show you some Democrats.
No, I'm not implying
Yeah. So here's community note. So let's make it like the community note tells us, and there's the actual plot. By the way, this one is even worse if you include the other years, because it just feels like this. It's basically constant and just going up and down. This is like random variation. Okay. All right, now sometimes when we're using, so now we're moving on, that's clear, right? So just know when to start. If you're using bar plots, you're implying you're using length as a visual cue, you have to start at 0. Now that doesn't mean you always start a plot at 0. If you're using position, you don't have to start at 0. Because position, we're not looking at how far it is from the axis. We are actually coming to the, sorry, the audience can't see me pointing. Maybe I should use the mouse. So with the, oh, they can't see me? Okay. So here you can see it's 79 and this one's whatever, 42. So we know what it is. There's no, so if you come up, you can say here, oh, here you're exaggerating the difference, but the mouse is better. On this side, you could say, oh, you're making it bigger than it is. But there's something here that there's some information here that is actually important. This is the this one is the life expectancy by country, by continent, and each point is a country. So what I want my audience to see when I make this plot is that there's differences across the continents. Right? You can see that by looking at like kind of the middle, but also there's variability. There's a lot of variability in the African continent, more than in any other continent. That's important information, I think. It's easier to see that here than here. This space is wasted if I want to just see like what's going on. So in this case, it's because I'm using position. You can use both are okay. It depends what you're trying to convey, but this is fine. Right? Again, it depends what you're trying to convey. All right, so there's another, this is also the White House, but in 2011. So another principle is that it relates to using the wrong visual cue, but also using it incorrectly. So here they're using the visual cue of, which one? Area, yeah? Okay, so if you just go by area, how much bigger is the American economy compared to the Chinese economy? It looks like it's like what, like 8, 9 times bigger? But look at the number. It isn't. It's 3 times bigger. I mean, if you want to do this one, that's like 10. How many times does that little circle fit there? And it's like 40, 30 times. So the problem in this plot is that they're using The area isn't, what's related to the number is the radius, not the area. So this is squaring the differences.
Actually, the 5.3 trillion for Japan looks like exactly the same as 5.7.
Yeah. So here on the right is how I would make the plot. That's area. Here I'm using area. I'm sorry. Here I'm using area to show the numbers. It's bigger. Yeah, US is bigger. Just show the right plot, Obama. Bigger. Gotta make it that much bigger. Okay, so that's another principle not to distort especially with area. I see this a lot. Now, however, with that said, why are we using area in the first place? Let's use the best. No, the best one is oh, what's going on here? It's all yellow. Okay. Yeah. You can use length, which is the better cube. All right. So that's one. So all right, I'm moving on. So stop if you have questions or comments, stop me. But I'm moving from different principles. All right. So the next one I have here is this is a quick one, and it's very useful for us in data analysis for cancer research because these kinds of plots come up a lot. And it's very simple one. It's the order categories by a meaningful bio. It's relevant to us because we go sometimes with what R gives us. So here's a plot. It's some random thing. This is murder rates by state in 2014, I think, or maybe gun murder rates. So
captions are also useful.
Oh yeah. Yeah, these ones I speak them. So yeah, I should have put a title on this one. We can go change the code really quick. Now it does have it here, right?
Yeah.
So the order is what R does with ggplot in this case. You see what it is? It's alphabetical. Why? What does that have to do with murder rates in alphabet? It doesn't matter. There's no reason to do that. So the defaults are often just kind of arbitrary, but there are reasons to order them in some meaningful way. And a good way to do it is by size, because then it just becomes easier to read and see patterns like, oh, these are the three biggest ones, right? It's not super easy to see that right away here. So this is just a minor thing. And I point it out because the defaults in R are not the best. I would prefer it if that was the default for the bar plot. What is this one called in ggplot? It's Guillaume call and flip. Anyways, it's in the code. Okay, so here it's also good for box plots. We're gonna get to box plots soon. I'm assuming people know what they are, but this is an example. If you look at the x-axis, this is GDP for region of the world. There's a lot of regions, and it's ordered by alphabetical order. No good reason. Now look at the one on the right. It's the same data. It's just a little easier to look at. That one's ordered by the median of the region. All right, so and then you can look at the code. There's one line that does that for you. I think it's like reorder or something like that. I don't know it off the top of my head, but it's in the code. All right, this is the biologist in the call. Listen to this one. Show the data. Important. There's many times where it's easy to. Sometimes you can't show it because there's a million points. Okay, but when there's 4, you can show it. So this plot needs to die. It's in all over Science, Nature, Cell, the dynamite plot, or has other names. It's like Cell again. It's like a default thing. It shows you so this is two sets of numbers, males and females, and this is our height in inches. So I have 2 populations. I'm using this as a very simple example, but you can think of so many other things. It's showing the mean and the standard error. And the standard error is shown as an antenna on the top of the building. You see it up there? These plots just look through the literature, they're all over and they should never be made or included. They are, there's showing, this plot is showing 4 numbers. Your printer is wasting all this ink for 4 numbers. And then I mean, you can just speak them or put them in a sentence. But more importantly, suppose you want to know, if you want to know something about these populations, like this is not a great example because we know what this is going to look like, but imagine you didn't. Imagine you didn't know how height is distributed across the population and you wanted to explain it to somebody. This plot is missing a very important part of it, and it's that there's a lot of variability across people, right? So that you want to show the data. Here's the data. It's just showing it. Now see, there's a lot of variability. I'm not starting at 0. That's okay. I just want to kind of see the numbers. Now this one is not a great plot, but it is showing you the data. I'm going to get to better versions very soon. Okay, so when you're making one of these, and again, if your PI makes you make it, you can still put the points there just so we can see where they are. This is very common. You have 3 mice and there's 3 points, but this is what you see instead. So this is not good because this is not great because there's about 1,000 points here. We don't quite see it. There's a lot of points in the middle there. That's an important thing about height. Most people are in like this between some standard deviations from the mean, 95% are within 2 standard deviations from the mean. So most points are here and here, and we don't really see that well. So there's something called jitter that lets you see it a little better. Right now we can see it better. There's more points here in the middle. You also see that there's, in this particular data set, there's more males and females. So jitter is a good thing to use when you have a lot of points. But you can also add summary things like box plots or histograms that are even better. So here's a histogram of the 2 heights. We went from those 2 block to this. This is so much more informative. All right, now this is gonna bring me to another principle. This plot isn't good. It's better than the first one, but it's not good. It has a couple of problems. See if anybody sees it. If I want to see I want to compare the 2 groups. There's 2 things I can do here that will make it a lot better. So think about it. Suppose you don't know males are taller than, on average, than females. Can you see it there? Is it obvious from that plot? It's not completely obvious. You kind of have to go down, draw a line, right? And then you would figure it out. So problem number 1, the axes are not the same. The x-axes are not the same. So then it's harder to see that the one on the right is shifted slightly. But also, you want to ease comparison. You also want to this is a principle. So let me just read it. If horizontal comparisons stack graphics vertically, that's what we want to do here. We want to see, we're comparing 2 things. We want to see where the middle is. You stack them vertically. If it's a vertical comparison like box plots, then you stack them horizontal. So here it is now, done right. Now it's immediate. I see. Males are on average high. There's a lot of variability. The range, you get all this information now. So that's
The axes are still.
yeah, no, it's coming now. Here. There it is with the axes the same. Okay. So this is another version here. It's we're comparing horizontal things, vertical things, so we stack them horizontally. So those 2, you can either, I've found with both of them, I slightly prefer the histogram in this case, but left is what you will see in journals all the time. Now, the information that's in that blue line, that's not in that blue line, look at how much more you get from the other plots. I'm starting to see people, huh?
You got it hard.
Oh, yeah. So yeah, please, if you can, I know it's hard. I know your PIs are stubborn and whatever, but this plot on the left is just so bad. It makes me sad every time I see it, and it's almost in every paper, including some that have me as a co-author. So if you can convince others to make one of these other versions, it's much better. Okay. All right. Choosing effective axes, axis ranges. This is very quick, but I see it a lot. But now that we have big data sets, I see this a lot, people, especially when you're exploring the data. Just this is good. Like, oh, I want to pick the range. So the default in R, and I'm assuming Python too, is that when you make a scatter plot, for example, it just picks the range of the points as the limits. That's a good default. It's a good default. It should be the default, but it's not always great. So for example, here, if I ask you, are these two variables related? Can you tell a little bit how much? Problem is 95, 90 more, 97% of this plot is white space with no information. All this space is only there so that we can see that this is an outlier. So now I just said show the data. Now I'm going to tell you don't show that point. So this is I'm going to be very careful here. This is for exploration. Right? So you're trying to understand your data. For a figure, we will have to be a little more careful maybe to figure, because we don't want to hide that outlier. But you can always say, we removed one point because we wanted to show the relationship between the rest of them. That's the same plot, removing the outlier. Right? So now we see something very clear. Right? There's some kind of a correlation with those 2 points. You can kind of tell it's probably like 0.8, 0.7, whatever it is. Right? So this is important. If you're exploring data, you might miss things if you just go through this and leave that point out. It's very common. I put it in the middle here because with high throughput data, it's so common to have like 1 or 2 points that are way out there, and you're making these plots, and then you might miss something really important because you never saw the pattern. I should put something more interesting here so it shows better. Okay. So this is more of an exploration thing more than a presentation plot. But again, you can still say, I removed an outlier that was this much, and here's the plot. Here's an example in real life, courtesy of Meredith Megan, of this error, of this problem. So here in this plot, if you squint and you look at this part on the right, they're showing you these confidence intervals, which is where the action is. That's really where you want to go pretty much immediately to see what's going on. And you want to see who's higher, who's lower, which one's cutting the center. But you're wasting, look at all this wasted space because there were these 2 cases where the confidence interval was just massive because I guess they had like very few data and it's a big, so, you don't get to see what's going on. I don't have the alternative here because it's from a paper. Next time I'll do it. But imagine if you could see more clearly. Here's another example. Like the range they're showing is from 1/100 to 100 odd percent change. Sorry, increase, multiplicative increase. So you really want to be seeing what's going on. Is that one touching? Is this one bigger? You can't see it because again, you've wasted all this space just to show this. And again, you don't want to hide. I'm not saying hide it. I'm saying for the purpose of showing what you want to show, you can say there was a couple of them that were really big, and here's the rest. Okay. This is related to wasting space, so transformations. So here's a terrible, terrible plot comparing continent population sizes. Give me a second. Those are wrong. So if I'm asked to show you how many people on different continents, I show you this plot. It looks like Asia is massive. It's not. I'm not showing the data. And there's another problem here. Let's show the data. There it is. There's all this. Just turn you off. Okay. So see all this wasted space? That's because, well, there's these 2 points. Anybody want to guess which countries those are? you can't guess that. You don't know population geography. That's India and China. This is they're not outliers, that's part of the data. But there's certain outcomes, population is definitely one of them, where the data is log normal or log normal-ish. It's closer to being log normal than normal. So it makes sense to look at log transform data. In genomics, we have this all the time. In counts, it's when the mean you have, well, it's not that it's counts. The distribution of gene expression, for example, tends to have a very long tail, log normal tail. So if you plot just the raw data, you'll be wasting all your plot on the biggest points, just like here. If you take a log here, it's a lot more, it's easier to see. Right? So I went from this terrible plot to something that basically shows you all you want to know. Now see how I have the axes very well labeled? You can either show the log or the numbers in a log scale. Either way, it's pretty clear what's going on. 1, 10, 100, 1,000. It's in the log scale, and now you can see what's going on. Actually, Africa has a higher median than Asia, but those two countries made it seem like Asia was this massively big in population-wise. Okay, so again, comparison, same data, two different plots. The one on the right shows you so much more. We did two things. We show the data, and we use a log transform. All right, so now getting a little bit more into finer things, finer points. So this again has to do with easing comparisons. So when you're trying to convey a message. So here, what I'm trying to convey is that this is comparing 1970 to 2020 GDP. And I'm trying to convey that the income gap between countries has actually reduced dramatically in the last 40 years. Big change. So it used to be that in the 70s, Western Europe and North America were very rich, and pretty much the rest of the world was very poor. So you have like these two separate things. But today, that's not the case anymore. I want to show that, and I want to see which of this is continents. It'll be better by region, but I'm going to do it by continent. Which continent? How did they improve? Which ones stayed behind? Which ones went forward? So I want to compare, say, let's suppose I want to compare Asia in the 70s to Asia in the 2010s. It's there, you can do it, but it's not super easy. So all this talking just to say, put them next to each other. Okay? They're next to each other. Now you can see it now. And also, since we're comparing two things, you can make it very clear what those two things are by adding color. So this plot's much better than the first. And there you can see, look at Europe. Didn't change much. This is inflation corrected, obviously. Europe didn't change much, but Asia certainly did. This is log scale. It's like, what is that, like 4x? So Asia went way up and actually almost caught up to Europe. The Americas also went up. Africa was, some countries went up, others, yes. But anyways, the point is, put things that you're comparing next to each other. This is another one you see, we have to do often.
What's with your y-axis?
What's wrong with it?
1 to 8 to 64.
Yeah. What's wrong with that? What do you want to see instead?
Log 2.
Huh?
Log 2.
Yeah, log 2. When it's log, it doesn't matter. What do you want to see instead?
Or round numbers, I guess.
Round? Those are all round numbers. Oh, you want to see like this one? That would be 32, and this one would be 4. That's it. I can add that. It's not. I think we can do it. We can do it in our heads. This is dollars per day, so it's 64 is very rich. $1 a day is very poor. It's better than the actual whatever. When they show you I don't know what. It's hard to convert. What does 20K mean exactly? So this is dollars per day. So it's my favorite way of showing income or GDP. dollars per day, on average, what a person makes or whatever in GDP wise. But what I'm still confused. What you want to see? Like, you want me to put 4 and 32?
No, you're saying like multiples of 10.
Now it's probably like
Yeah, if it's a
100,
log
100,
axis, usually it's 1 and 10.
Well, that's just that. So then you want to see 1,
and
10, and then that's it. 100 would be up here. Okay, so maybe we'll talk about that. So
that would
log when you're logging, good that you raise this. When you're logging, so in math, let me start by saying that you should never, well, I shouldn't say never, but I would be surprised if you gave me an example with data, you have a reason to use natural log. E is a number that comes up in math. It's super important, and we use it in many equations, including the normal distribution. But when you log something, it doesn't really matter what the base is. The point is it converts multiplicative to additive, no matter what the base. So when you're plotting, especially for yourself, you want to use a base that you can translate to the original number quickly. So 2, I have it memorized. 2, 4, 8, 16, 32, 64, 128, 256, 512, 124, 428. Keep going, right? 10, I have memorized. That one's easy. 10, 100, 1,000, 10,000. E, 2.7, and then what? What's e to the 2? So you should never use E, because we want to be able to look at the number, convert it in our heads. Now, when to use 10 versus when, so 2 and 10 are the ones I use. I don't go anything else. 64, what is that, like astronomers maybe use that? 2 or 10, because I can do it in my head. Which one? Here I use 2, because if I had used 10, okay, I want to have as many single digit numbers as possible is what I'm going for. Because single digits are just easier to put there and look at. And so this one gives me 1. I actually would have been a better plot with 4, 8, 16, and 32. 16. So this is, yeah, it's in 4. So 4, 8, 32, 64. Am I doing that right? Maybe I'm missing 1. 4, 8, 16. Yeah. So if I had used 10, then I would have had 10 would have been here, and then 100. No, it's nowhere. And then I have to use like decimals. So that's how I do. So in genomics, it's almost always better to use log 2, because the range is about 0 to 16,000. So that's 2. For population, where you go from countries with 10,000 people to countries with 1 billion people, definitely 10 is better. That makes sense? So that's how I decide. It's just I want to have round numbers at the end as much as possible. Sorry, as many single digit numbers as possible. All right.
Sorry.
Yeah.
So the only thing that is distinguishing 1970 from 2010 is color.
Mm-hmm.
So did you use colors that are distinguishable by people who are colorblind?
No, I have a slide about that. I think these are. So I use the default ggplot. The default ggplot isn't the best, and I haven't been great at improving that, but because you have to actually do work. But they're not they're not bad. I think this is maybe someone. There's 100 people watching them. There's probably somebody colorblind that can tell us. I think these are kind of distinguished. Anybody colorblind that doesn't mind? Okay, anyways. All right, so there we go. Next slide. So 1 in 12, 8%, and 1 in 200 women are colorblind. So there's all these palettes that you can use. Here's the one that ggplot recommends. Sorry, that it's an easy one to add. Forget the name, but you can look at the code. That one apparently is very colorblind.
Oh, Hito.
Okay. So I say this, but then I don't use it. So I'm guilty as charged. All right, now we're moving to 2 variables. So let's switch our brains to that. So with two variables, the scatter plot is pretty much almost always the winner. But there's a couple of exceptions. Well, actually, bland Ackland are also usually very good. And then there's slope chart. So I'm going to give you two examples of other alternatives. So here's the first one. It's called the slope chart. It's a good plot. When you don't have too many points, when slope charts got a little fashionable at some point and everybody was making them, and then it was like plots with 1,000 points with all these lines going all over the place and you couldn't follow them along. Not a good choice. But with this plot, which is just whatever, 8, 10 countries, it's good. So this is I'm going to say when it's good. So this is comparing life expectancy in 2010, 2015 by countries, by sets. I just picked these countries. And I want to know which countries, I want to know two things. Which countries have higher life expectancy, and also which countries are getting better in these 5 years. Right? So I want to know two things. So what are the visual cues? Now a quiz. What visual cues am I using here that help me show two things in one plot? I'm using two visual cues. Slope is one. That's the harder one to see. The other one's obvious, is position. Right? Position. Position. Oh, sorry. There's the position, and then there's the slope. The slope tells me how much better they're getting. So that's why this is a nice plot. The angle, sorry, not the slope, the angle is the other visual cue. So the higher the angle, the more the country improved. All right. So that's what this is a good plot for that. Now, here's the scatter plot version, so it's a little bit harder to see which ones got better. Right? They're all above the line, so they all got a little better. But how the distance to that line is what tells you how much better they got. And that's like tilt your head and kind of like, so it's not as good. But this brings me to my next plot. This one is actually in genomics in particular. It's I would say probably more common than not that you want to make a bland outline plot over a scatter plot. And that's because if what we care about is the difference, if that's what our main interest is. So for example, if your main interest is the log ratio of the expression of two genes, then you're plotting genes, a scatter plot of the expression, then that log function is like this 45-degree distance from the horizontal, from the identity line. It's fine, you can see it, but it's what the bland outline plot does is it puts what you care about on the y-axis. These ones are also called MA plots in genomics, but this is the original name. So this is the same plot. It's similar to a 45-degree rotation. It's the same information, but now I have the difference in the y-axis and the average on the x-axis. So now it becomes immediately obvious that Portugal is the country that the most improved. It's not that obvious from this right there. It's far, but it's not immediately obvious that it's farther than, for example, Spain. So that's what that one does. It just puts the difference on the y-axis and the average on the x-axis. If you know linear algebra, you know that that's a 45-degree rotation. Then you need a square root of 2 to make it exactly right, but basically you tilt it like that. So it's not you're not removing or adding information. You're just changing what transformation of the data you look at. Very simple, very powerful, especially in genomics data where you see patterns. Sometimes you see a scatter plot. So this is very common in genomics. I should add that example here, where they show a scatter plot and they say, look at how good the replication is, and the correlation's like 0.9. Looks like it's really good, but then when you look at the MA plot or the black outline plot, it has all these weird curves and it's fanning at the bottom. It's just things you don't see when you make the scatter, because the thing you care about most, which is how alike they are, the difference is not shown in an axis. Okay, I'm gonna add that for the next time. The examples of that. Yeah, so just let me say just one more thing about that. So to be very precise, if you're giving two replicates of gene expression data, whatever else, or gypsy binding size or whatever it is, relation values, and you have two, they're replicates, and you want to see how similar they are, make a scatter plot. It's not the most powerful way to find problems. Instead, you do log ratio on the y-axis, and you do average of the log intensity on the x-axis. And you'll see a lot more. Okay. Now we go to third variables. Now we've done two, now we're going to go to third. So when we do three, now we're going to not be able to use our favorite cues. Position, length is gone once you go to, well, not completely gone, but you can have lengths of two things next to each other, but that's not great. It's better to make a scatter plot. So now we go to third. What can we use? Because we can't have three dimensions as I'm about to show. So wait, let me see when I say that. Okay, it's coming up. So I'm going to show later 3D, pseudo 3D is not good. I'm going to show you examples of how bad it can be. So what can we use to encode a third variable? So now we have all the other cues left, area, brightness, hue. So here's an example with a lot of information in one plot. This is again, I'm using this example a lot. So this is infant survival proportion. This is from Gapminder. It has all this information. Data set. Infant survival, this is per country, and then dollars per day GDP on the y-axis. And the y-axis might be in a logistic transformation. So what you see, there's definitely a relationship there, but that's not the point. What I'm saying is I want to show other things. I want to show what region of the world they're in. That's a categorical variable, so I'm going to use color. So the color showing that. I want to show how big the country is. So for that, I use area. You can see this is that's China, and this is India, right? The two big circles. Then I have one more thing. I just wanted to pick an example: OPEC or not OPEC, oil producers or not. So I use a symbol. And so I also have that in R. I have that very easily available. These are the point shapes in R. You can distinguish things with shapes as well. Okay, so now when we're going to use color to show numbers. So in that plot, we didn't show, this was showing category. Category is easy, right? Each category has its own color, no problem. But what do we want to show number? So there's two choices you want to make here. It's an important difference. So there's two kinds of there's a distinction we make when we want to show numbers. There's when we're showing numbers that go from low to high. Those are called sequentials. And there's palettes that are good for that. This all comes from psychology studies. So sequential colors, these are palettes that have been determined by psychologists that are good. And they go from a low value to a high value. So you have blue, very, very light blue, to very, very dark, for example. There's other you can pick. This is from the Art Color Brewer package, which is still great. They have all these palettes, right? So there's another one. The other one is diverging colors. That's when there's a center. And then there's temperature, right? So there's average temperature, and then there's high temperature and lower than average temperature. As opposed to like counts, numbers of this, numbers of that. That's sequential. There's other palettes for that. So I like using the it's the one that goes from red to blue. Yeah, is it this one? This one. That's one I like this one. So it goes from red to like white, and then it starts going up to blue. This is an important distinction. It doesn't make a difference. So if you have a center and you want to see things that are deviating from that center, you want a diverging palette. If it's something that's just growing, you want a sequential. All right, another one that you still see, again, it's mostly probably Excel's fault. So pseudo 3D, no good. So we can't even, humans don't really see in 3D. This is why it's hard to parallel park. I think Tukey said we saw in like 2.4 dimensions, John Tukey. But pseudo 3D is just even worse. So I'm just showing you this by an example. Don't use pseudo 3D. I'm going to show you alternatives. So this is an example. So I'm showing you three curves. There's just three curves of 2 dimensions. Those and proportion survived. Mice and the Shan's right. So I'm going to ask you when or if the purple line intersects the red or the yellow. Can you tell that plot? Because they do.
Exactly.
That's the plot I would make, and it tells a completely different story. First of all, it's very easy to see when they intersect. But also you just see better what's where. There's no point. This is actually just gratuitous 3D. There's no reason to. There's not even the third dimension doesn't even do anything because you're already using color to this thing when you put 3 graphs. So here's another one is 3D pie charts. Still see them every once in a while. It's like you take something that's already bad and you make it 3D, so now it's even harder to tell things apart. Or this is another one, like how high is this? How tall is this bar? Is it 25? I guess maybe 25. It's like, why not just make it without the 3D? You see it right away. So in general, avoid pseudo 3D. No reason to do it. There's always some a better way. Even those ones where they show PCA, there's 3 PCs. It's impossible to see it. Don't make those. If you when hologram technology comes around, maybe we can start doing 3D. But for now, there's no point. It's better to show scatter plots. If you're showing 3D, 3 things, you can show each the scatter plot of each pair of dimensions. Okay. Before this is almost I'm gonna there's one more thing I'm gonna show it's very specific, which are UMAP plots, why they're so bad. But before I do that, I wanna just add this. This is related to visualization because often we use tables to show data. It's not really visualization, but I just want to point this out because it drives me crazy. Don't copy and paste what comes out of R or state or SAS or whatever. Those significant numbers, just think about them. You learn this when you learn this in high school or even before. How many significant numbers we should show? This is just there's nothing there, no information out there. The standard error is way bigger than that. So just make sure that you pick good round it in the right way. That there you have to think. Each case will be different, but look at that table, compare to that. Look how much easier it is to read. And there's another principle about tables, which is a little subtle. I don't know if there's studies for this, but it's certainly true for me. To me, it's much easier to make comparisons going vertical. So when whatever you want to compare, put it going down. So here I want to compare years. I want to see that the measles rates were going down through the years. And I want to see it, I'm seeing it for 3 different diseases, right? So because I want to see it, I want to compare it per year, a plot would be better. But if we're making a table, it's much easier to see. It's not a huge difference, but it is, for me at least, it's preferable. Okay. Also, there are the functions you want to use in case you don't know them. And then you can also use, this is in R, you can do this. You just want to just stick to 3 significant digits throughout the whole R session. All right, so here it is. So I see sideways, I don't know if others have that problem, but it's just easier, going down for some reason, easier to make the comparison. So this is the other version of the table going the other way. Yeah, so there you go. I don't know. Maybe others don't have that issue, but I do.
Well, the digits are aligned.
Yeah, there's all different, yeah. Something like that. You can see if the digits are getting smaller, you can see this shape. Okay, so before I go on to my last very more specific uh, uh, very timely topic, I want to just remind you I gave you all these data visualizations principle, but if you're a data analyst, there's different audiences when for plots. And the big distinction is you are your audience most of the time. You're making plots to find problems, to make discoveries, to check, to do diagnostics. That's very different than when you're showing the plot to others. So just keep that in mind, that there's a difference. Often you see people giving presentations and they show the plots that are for me, for them, and it's just not pretty. No, maybe I was a little guilty of that today because I just ran code and I didn't make it pretty for you guys. But when you're showing it to others, you want the labels to be there, you want the labels to be readable, all that. It's important to do. And then the other important distinction is when you're conveying, if you're giving a talk to experts or to very general audience, to donors or some colleagues that maybe are quantitatively challenged, you want to think about that because things like the log scale, which might be the clear best choice for 2, sometimes is not the best choice for 3. The log is what percent of the population understands it? Maybe 5. So just keep that in mind. Yeah, so again, when you're making the principles are apply across, but then there's the details that you want to keep in mind who is the plot for. All right, so my last topic is about UMAP because it's just so ubiquitous. And I just want this is now we can use what we've learned to see this why are we making this plot. This is a plot from a paper. What are we seeing there? What are we so what are they're using position. What is position? UMAP dimension 1, UMAP dimension 2, and what is that? It's not a rhetorical question, but you want to know what the answer is. And what does it mean to be for 2 points to be here and there? What's that? Does that have a meaning here? That's definitely pretty. But what are we showing? What information? So first, let me say why UMAPs are great. They're useful in the right context. I'm going to explain what that is, and then I'm going to tell you why the way they're being used today is just adding confusion. So what do they do? First, let's suppose that you have a data set with high dimension. That's when they're useful. So here's an example. This is a very classic data set. It's the Iris data set. It's flowers. It's very boring. It's been used for too long, but it's still as good. It's a good example. So this has 4 dimensions. So with 4 dimensions, if I want to explore the data and see what's going on, I can plot it. Right? I can plot each pair, for example. This is one thing I can do. There it is. That's all the dimensions plotted against each other, and I start seeing things right away. I can look at sepal width and petal width. There's some information there. It turns out that's like the 2. This is like one a species, and these are the other 2 species. Here too, start seeing stuff. All right, and you also see that these 2 are very correlated, they're not adding information. Maybe we can get rid of one of them. Point is, scatter plots, good. Now, what about when I have 784 dimensions? So this one, no labels. Sorry, this one is. This data set has 784 dimensions for something like 1,000 observations. What are these? This is data from the very famous machine learning data set, MNIST, which are digits. So images of digits. So you have, in the machine learning class, we did this. We used this data set, so I brought it back. So you have 1,000 observations with 784 predictors. You are not going to make a pairs plot for this. So how do I start getting a sense for what's going on? So one of the things I could ask myself is there enough information in the data to separate out the digits? If there is, UMAP will be able to do it. We'll be able to show it in 2, almost always. So here you can see it. So the labels are the digits. So it goes from 0 to 9. And when I look at the first 2 dimensions of the UMAP transformation, it's almost there. I mean, these 2 are hard to tell apart, but this is kind of split. There's a little bit of agreement here, but still, it's just 2 dimensions. I mean, we can split them perfectly, almost perfectly with 35 dimensions, but if we just want to visualize it quickly, I make this plot and I know, okay, I can do something with this data set. Now let me go find how to split. That's it. Exploration. This is not in the paper. It's not going to be in the paper. There's no reason to put it there. I want to actually build a machine learning algorithm that can do it. 99.9 accurate. This is not going to help me make that point. But it did originally very quickly. Made UMAP. Okay, I'm going to be able to do something. All right. Now, so that's one of the good uses for. So we're single cell. Sometimes you can say, wait, is this? I know there's these different cell types. If a UMAP can't split them, there's a problem with the data. Right? So now if it splits them, it doesn't mean there's no problems with the data, but if it only goes one way. If UMAP can't split them, there's probably something wrong. But if you see splits in data, and I'm going to show you an example, it doesn't necessarily mean there's real difference in the data. That's the problem. It goes one way. It doesn't go the other. So here's an example. This is a UMAP plot of multivariate normal data, just randomly generated multivariate normal data. I think I can't remember, 10 dimensions. There is no clusters. It's just multivariate normal data. No clusters. UMAP tells me there's 2 clusters. If I interpret it incorrectly as it's being done right now. This is the simplest example of something that's obviously not clustered. But there you go. Now, if I do it again, now it's not clustered. Right? So there's also this random component that isn't really explored. So I generated the data again, I do it again, and now I get this. All right, so I'm going to show you, just to motivate it more, an example where it actually led to incorrect interpretations, especially if lay people are reading it. This is a UMAP of genotypes for human populations, self-reported. This is the all of us data set. All right, so they took the genotypes for self-reported Asian, Black, or African American. You can see the legend, right? There's the colors. They do a UMAP. Why? Because it's fashionable and it's kind of pretty. Because otherwise, what are they doing this? What do they want to show with this plot? What it ends up showing is that the blue group seems to be very far away from everybody else. Right? The orange group and the blue group are really different, and then there's just it's like these oranges that are here and not there. What does that mean? What are we supposed to make of this? I don't understand. I read the paper, I just don't get it. And it seems like it was just because fashionable and pretty. So let's suppose we wanted to show something about this data, genetics, predicting the self-reported race. So I'm going to focus on a specific group, and now all of us data is not quite public. and it's very hard to work with. So I went back to the one genetics data set that is really publicly available, the 10,000 Genomes Project, where you can get the genotypes. And I ran UMAP on 5 groups. These aren't, I don't think these are self-reported. I think these are groups where they went to the place. So these are British, that's red, Spain and Portugal, that's yellow, gold, I'm not good with color names. And then there's Yoruba, that's Nigeria region, that's blue. And then there's African Americans and Puerto Ricans. And you make this plot with UMAP and a story comes up. This is a story of Black and Hispanic and white somehow mixed with the Hispanic. That's the story that this tells. But and they're very different. Look how far they are from each other. But then we look at these 4 Puerto Ricans, what are they doing over there? And what is this African American doing over here? Is it that it was reported wrong? No, it's because that is, well, I'm going to get to it, but here's Europe versus West Africa. That's different. That's clear. That's not UMAP. That's PCA. And I can go and look at the SNPs that do it. Very clear separation. And I can look at the data and I can see why it is. Here is those, now I'm bringing back everything in PCA, and you can see there's no separation. There's no like split. And those supposedly wrongly classified people, they're all here in the middle. They have ancestry from Europe and ancestry from Africa, West Africa, and they're in the middle somewhere. And those are interpretable values. You can actually go and see the SNPs and see, oh, this person's probably half this, half that, or 1/3 this and 1/3 that from this plot. This is PCA. So the same data, PCA, very clear continuum of the admixture populations versus this total nonsense plot. It's not, and I shouldn't say it's not that it's nonsense. It's UMAP is doing what it's doing, but the separation isn't, it should not be interpreted. It's not meant for this. So with that said, so here's a better plot if you want to see what's going on. Here is the first PC. That one I can look at. I can see it's a linear combination of a certain number of SNPs. And yeah, there's Spain, and there's West Africa, and there's Britain, there's West Africa, and these guys are some kind of mix of these two, and these guys are some kind of mix of those two. And you can see that there's some overlap. Why wouldn't there be overlap? All right, so this is, to finish the talk, this is a UMAP plot of randomly generated, only one cluster cells. Now they have different coverage, and there's gene correlation. This is what happens when you put it through Seurat and you get clusters called, and then you make a UMAP plot. The message we should be conveying here is there is one cluster. That's the reality. And if you just run that without understanding what's going on, there you go. Put it in a paper. You discovered whatever it is, 20 new cell types from a randomly generated random data set with no clusters in it. So if you don't think that's happening and with whatever others are doing and publishing it in all these fancy journals, I would be very surprised. So be careful with this. It's useful, and if you want to use it, understand what it does so you can interpret it correctly. All right, I'm going to finish there.
Any questions?
Yeah.
There's clusters and sometimes there's not. Is it related to sample size? I mean,
Yeah, no, sure.
enough sample size.
I think if you increase the sample size, you will not see clusters. Yeah, that's correct.
Another question is about the PC.
Mhm.
I feel it's sometimes it's really hard to explain to non-statistician that what PC1 means.
Okay,
It's a linear combination of steps, but what is a linear combination of steps?
well, you can look at, you can,
Yeah, I can look at loadings. I mean, this is the most contributing loadings, but you.
oh, because they all get some. Is what you're saying. Yeah, so yes, it's true. It's hard to explain. It's not as hard to explain as UMAP, not even close. But it's not straightforward. Topic NMF is a little easier to explain, and actually NMF works I think even better than PCA in this example. NMF would be easier, right? That one, because then you get actual zeros and no, it's not easy to explain PCA. It's a difficult concept. That's what can we do? It is difficult. Some things are complicated. Yeah. Any questions? Any questions from the
Nobody's typed anything in chat.
okay.
Question, I don't really use UMaps in my work. What are you supposed to gain from like the map plot that you're showing?
Yeah, a pretty picture. That no, it's
What is it
not.
supposed to tell you?
So what the way it's misinterpreted is, so this is in single cell. You heard of single cell RNA-seq. So there's many ways, but some of the experiments they claim to discover new cell types. Okay. So they're saying to prove it, they show you this and say, see, that's my new cell type. You see how it separates from the rest?
Yeah,
But
there's a lot of like interpretation of distance as relatedness of one cell to another, but it's like.
So okay, so that's another issue that in PCA. So, but that's actually there's other I can give you other side, but that's a big one. But what's happened is it's become so used that it's almost like it seemed like maybe the editors are saying, where's your UMAP plot? Like if you write a paper in that technology and you don't have a UMAP plot, they're gonna say, where's the UMAP? So then people make the UMAP plot and put it there. Right? And it will look, it always looks pretty. So now getting back to this thing about distance. That's right. So in PCA, if the first two PCs explain 100% of the variation, then that means mathematically, that means that the distance you see, the distance you see in the plot, you take a ruler and you measure it, is the actual distance between the two high dimensional points. That makes sense, right? So if it's 80%, then it's an approximation. So the PCA plot has that interpretation. It depends on how much variability you explain, but the distance, you can kind of say that these two people are close to each other genetically compared to this person and these people. Right? It'll be a pretty okay approximation. With UMAP, there's none of that. That's not there. But except for what I said earlier, if you have things that are actual clusters, this is the example I gave with it. Let me just go back to it because I don't want to trash UMAP. I'm complaining about how it's used today in biology, but it's very powerful. If PCA wouldn't even get close to a plot like this if you show the first two. If you show the first two PCAs of the digits, you'll kind of see them, but they're all kind of mixed together. UMAP is actively trying to separate clusters in 2 dimensions. That's what it's built for. So what do you think happens when there's a slight little error that makes it seem like they're different? It separates. But when it's actually there, it'll do it. So it's useful to see that. It's useful to say, look, there's definitely something. Looks like there's enough signal in this data that can separate what I want to separate into groups. Now I have to go and find what it is. Which dimensions are there? Is there some algorithm that will help me do it? See what I mean? But again, it's going from 784 to 2 and showing you the separation, which is pretty cool.
So it's better to like confirm the cluster you already know is there rather than like discover a new.
I wouldn't use this for clustering. That if you're then gonna cluster, then that becomes a separate problem. I want to see. You can run if you run hierarchical clustering. I think a lot this kind of comes out too. So that would be another thing you could do. You can run hierarchical clustering and then look at it, but that's a tough plot to look at. A hierarchical plot of this would have 1,000 leads. It's not as easy to look at, but it has a similar information because you'll be able to see that the numbers at some point can be split. Yeah. So yeah, you could run a clustering algorithm and just say, look, they split. There's no plot. So this is the way. This is one of the reasons it's nice because then the people use it. It looks nice because if you show the clusters like this one is showing, in this one the colors. I didn't say this because I assume people knew what Seurat does. The colors are obtained from clustering, not from UMAP. They're added to the plot, and of course, things that cluster together are going to clump together in the UMAP plot. So they show up like the colors look like they're. All right, I think I saw somebody raise their hand on the corner. No?
We do have a question on the chat.
What is it?
Any comments on things to consider when trying to make plots readable for you, the analyst, on your laptop screen versus readable for a viewer of a presentation for things like color contrast or large font sizes?
Yeah, definitely font size is the main one. That's the one that the ones that you make for yourself are never going to have the font size you want for the paper or the talk. The main suggestion is make the fonts big enough so that a older person in the back can see it. Someone with glasses or whatever can see it. So that's usually much bigger than the default. Now the other thing that changes is the aspect ratio. Might change, but because if you're looking at it, you might be you have the option to look at it in any kind of aspect ratio that's useful. In the presentation, it's almost always better to have it follow the aspect ratio of the talk, which is whatever this is, 16:9. But sometimes you can't do that. Sometimes it has to be the other way. It's just better. So that's more of an artistic choice. Let's make it look good. But that's the main one, is that you want the fonts to be readable. And that's a big mistake I see very often in talks by statisticians and computational biologists, that they make the plot using, which is good. Definitely use R Python to make your plot. If you can avoid Adobe Illustrator, do. Try to make it all reproducible. But the defaults sometimes make small for presentations. Yeah, that's another thing. Maybe we should include that in my talk. How, if you can, that's more a reproducible research workshop, that Adobe Illustrator is not reproducible. Makes me very nervous when I see people using it for scientific papers, because points can move when they're not supposed to. Yeah.
There actually are some packages that are really hard to control, like labels. For instance, I sometimes use the artworks that are splitting trees, and to put labels on each of the branch, that's like insane.
It's hard, but yeah, ChatGPT should,
And
whatever you are using.
yeah, I try that too, but sometimes it's hard. So that's probably.
Well, that one you can maybe draw. That one's okay to draw.
And Adobe Illustrator.
Yeah, or
Illustrator
PowerPoint.
case, yeah, because it's really hard to modify them within the code to make them.
So that one is, yeah, because that's not really a plot. It's like a schematic.
No, it's not really a plot. It's something that is very useful, and I found that the PIs usually are happy with, because they.
Yeah, I think that's fine.
But the labels are really nice.
Changing the labels for one of those cards, I think. Right. Question, everybody's gone.
Basically, the whole thing was not, don't use Excel for.
Oh, no, that was what I was saying.
Excel.
Is anybody using, I guess, maybe?
No, I hope not. It's there.
When I started, a lot of biologists used
Oh,
Excel. Now I don't think.
That's.
A lot of journals require like supplementary tables.
Tables.
If it's a table, but it still risks the gene symbol problem and.
But whatever you submitted to CSV is considered an Excel file
No, but for the plots, I meant.
for the plots.
I'm also guilty of teaching Excel for plotting. That's something.
Oh, I don't even know how to do
They
it.
made me do it. Wasn't me.
There's an Excel, and there's this great package called GG themes or something. There's the themes. What's that one called? I think when we did the GG plot workshop, I think I talked about it. It gives you ways to make your plots look like Economist or 538. There's an Excel one.
Yeah, no.
That's how I made these.
No.
I didn't make these in
Okay.
Excel. You think I
Those
went
were
to
not-
Excel?
Those were not real Excel? Oh, come on.
Well, I mean,
We have the 3 days straight.
this one, no, I made this with ggplot. The Excel theme.
Excel package.
Kidding
me, man? I learned Excel just to make this thing. Now, these ones I think are Excel. These ones I didn't.
The 3D, yeah, that's
From a paper.
Excel.
Those are
Also-
probably Excel.
Yeah.
Yeah, I hope R doesn't connect that you made that.
Yeah.
You know, in R, have you looked at the help file for pie chart in R? It basically says don't use this. We've lost that battle. I've embraced pie charts because it's like, what are you gonna do? Let's try to make them as best as possible, but there's still some situations where, you know, where I've seen them use it, I think it's okay. It's when they have like, there's X and a Y, and then each XY has some proportion associated with it. So there's like a little pie chart in each one. Because that would be, but how else would you do that?
Yeah.
I think that's okay. I think that's fine. I mean, it's two pie charts with two things. That's when it kind of works.
Yeah,
Because you immediately know
Yeah, that's it's a distinct.
with just two things. So I think that one's. Yeah. All right. I also have, let me see if this just shows up. Oh, here we go. This is the other use of a pie chart.
Right.
I made that in R.
No.
Base R.
No.
All right.
All right,
Good.
thank you very much, Rafa.